Yeah, thank you very much for inviting me to give this talk. Yeah, it's an honor. I'm excited to talk about this stuff. So what I'm going to talk about is something that my student and I and a collaborator and I have worked on, and which we call as dynamic homeostasis. And we're going to specifically look at relaxation and bursting oscillations. Um, so first, whoa, okay. First, homeostasis. So what is it? Homeostasis, broadly, when someone thinks of homeostasis, thinks of a system where you have a stable internal condition, like a human body, for example, amidst a variety of external factors. And the idea of homeostasis is sort of ubiquitous throughout both theoretical and even clinical physiology. And biologists and physiologists list homeostasis as one of the top three tenets that are fundamental to their field. Like they say, you must absolutely know homeostasis in order to be sort of considered competent as a physiologist or something. Um, but if you actually look through the literature, there's no really consistent definition for what exactly homeostasis is. So here's a set of definitions that I compiled from some of the undergraduate physiology books and even some medical school books. So you have the first one, stable internal state with respect to external variables. That sort of coincides with the general view of what homeostasis is. But then in the principles of human physiology, they add this wrinkle that it's a system that maintains the internal stability. So there's this idea that homeostasis is some sort of a thing that conducts actions that maintain internal stability, which is kind of not intuitive or not typical. Um, Textbook of medical physiology, which is one of the really big ones, maintenance of nearly constant conditions in the internal environment. So then there's this wrinkle of nearly constant. Keeping all internal conditions fixed regardless of internal external changes. That's an undergraduate physiology book. And then my favorite, you know it when you see it, in the textbook of physiology by Fulton. So there's kind of a range of ideas on how to define homeostasis physiologically. Um, and so there's no really consistent definition. Uh, And so here's some examples that you can think about for homeostasis. So human body temperature is a typical example. Whenever you go to the doctor and they measure your temperature, they're looking for something like 98.6 degrees Fahrenheit, is typically what they want to see when they measure your temperature. Blood glucose levels for a non-diabetic person should be around 85 milligrams per deciliter. This is a number that sort of is a benchmark for if your sugars are in control or not. And in general, the idea is that hormone levels should not fluctuate. And so there's all these ideas of a if you think about it from a dynamical systems perspective, these can be thought of as like equilibria of some complicated dynamical system. And so you can think there's like some sort of a control system that feeds into an internal environment, and then there's a feedback loop, some sort of an error detector, which kind of pulls any fluctuations back towards a fixed equilibrium. And with this sort of control system idea, there have been several well-regarded mathematicians who have developed some of the network motifs that allow for a system that facilitate a system's ability to maintain a sort of a constant equilibrium in the face of external fluctuations. So these are some of the canonical figures: Mike Reed, Janet Best, Marty Golubotsky, and Stewart and Frederick. So they've published several papers and on how negative feedback can help support homeostasis. And so this is a what they call as a paradigmatic representation of homeostasis. So Z here is some output of some gene network, say, and I on the bottom is an external input. And notice that over a range of I from about 100 to about 250 units, the equilibrium does not change that drastically. And so they refer to that region where you don't have that much of a change in the equilibrium as a homeostatic region. And in particular, they call this a chair curve because you can think of the homeostatic region as the seat of a chair, and then you're kind of leaning back on the upper part, and then you can recline your legs, and so it's like a reclining chair. And so they say these chair curves are a one way to think about homeostasis mathematically. Um, but if we go back and revisit those sort of examples I was giving you about homeostasis, human body temperature should be 98.6 degrees Fahrenheit. But if you actually measure temperature throughout the day, it oscillates quite regularly. So this is some data from a paper. Uh, so it moves about one degree up. So it has an amplitude of about 1 degree. Sorry, no, like half a degree Celsius over the course of a day. Um, so that's your human body temperature. Blood glucose levels are known to have these ultradian oscillations, and so these things are also oscillatory. But we kind of say that the region at which these glucose levels should remain is around 85 milligrams per deciliter. And even hormone levels fluctuate quite drastically. And so the idea that all these things should be in homeostasis uh, is maybe a correct one and necessary for like proper physiological function, biological function. But the idea that they reach some unchanging equilibrium needs to be revisited, and we need to sort of broaden our idea of what homeostasis is. And so we need to understand in particular how stability or homeostasis can be encoded in oscillations. One very obvious way you can think about it is, well, what they're just doing is they're just looking at a temporal average. They're just measuring the temporal average of this of an oscillation, say, and they're aware of the oscillation. Um, they're measuring the temporal average and saying that's what we're thinking of as homeostasis. But oftentimes, when you have a sort of a high dimensional system, um, you have to ask the question of what do you actually measure the temporal average of which variable are you actually looking at? So that is a question that we'll look at in this talk today. So I'm going to start off by thinking about the Fitzhugh-Nagumo model. So Fitzhugh-Nagumo were developed by Fitzhugh and Nagumo. Um, as a simplified version of the classical Hodgkin-Huxley equations. And the van der Pol oscillator is a special case of these equations. And so you have a fast variable that encodes the membrane potential, and then you have a slow variable that encodes the recovery variable, so that affects the repolarization of a neuron, for example. Um, so J in this equation is an input to the slow variable. So you can think of it as some protein prefactors that are affecting the ion channel's ability to repolarize. And then mu is a parameter that controls the timescale separation. So mu is very large. So x is fast, y is slow. And alpha is a feedback strength parameter. One of the main features of the Fitzhugh-Nagumo model is that it exhibits excitability. So in panel A, I'm showing the phase space of the system and with the null lines of the system. So you have this cubic null line and this linear null line. And when the linear null line intersects the cubic along sort of the left branch or the right branch of the cubic, you get a stable equilibrium that is excitable, meaning if you perturb away from the stable equilibrium, but the perturbation does not push you past this middle branch, then I don't know if you can see my cursor, but if you get pushed away and you're not pushed past the middle branch of the cubic null line, what you'll do is you'll just rapidly equilibrate back to the stable equilibrium. But if you get perturbed away to the square, you've crossed that middle branch, and now you're excitable. You have to sort of take this huge detour to come back. to the stable equilibrium. So that's a sort of a canonical, a typical behavior of the Fitzhugh-Nagumo model. Okay. Um, but what happens when the line intersects the cubic null line along the middle branch? Well, it destabilizes, and you have a subcritical Hopf bifurcation. So you immediately get this sort of large amplitude oscillation, and you can see the time series on the right. Um, and we term this a relaxation oscillation because you're characterized with a slow buildup along the cubic null line. And then once you reach sort of the knees of the cubic null line, you shoot away to the other knee. And then you have this separation of time scales manifest in the oscillations. Um, and so our claim is that the Fitzhugh-Nagumo relaxation oscillator encodes homeostasis in the temporal average. So let's see what I mean by this. So as I increase J, this line is going to move to the left. So I'm sort of hovering around, I'm still intersecting the middle branch, and I'm still hovering around it. But as I increase J, notice that the oscillations kind of move. They have this maybe phase shift. But overall, the temporal average shown in the dashed line doesn't vary all that much. Um, and this is true in general across an intersection of the linear null line across the middle branch, across the entire middle branch, is that the oscillations will have this sort of phase shift, but not a significant change in the temporal average. And this is of the slow variable. And so here is a bunch of uh, points here. So on the right in panel B, I'm showing the intersection points of a linear nullcline as I vary J with the cubic nullcline. So this red is like a stable equilibrium, and that corresponds to a temporal average in the panel A of about negative zero point 0.4. And then if I move to this green open circle, I have a unstable equilibrium. And the temporal average affiliated with that intersection point is along this seat of the chair, if you will. So I have this reemergence of the chair curve where for all values of J where I have this oscillation, the temporal average of the slow variable doesn't really change all that much, which is pretty cool. So that was one of our key points. And then you might ask, well, what about the fast variable? You have the in the phase space at least, the oscillations, the limit cycles are sitting right on top of each other. So why can't you measure the fast variable to observe this homeostasis? The trajectories approximately overlap. So is the time average invariant? This might be a question that you're asking. It turns out it's not, because in the fast variable, the duty cycle changes as you vary J, and that's not what happens in the slow variable. And so you can see on the left-hand side, if you vary J and then you plot the temporal average of the fast variable, you don't get a chair curve, or you get a bad chair, a chair that you can't sit on really comfortably. And so dynamic homeostasis or homeostasis is not really encoded in the temporal average of the fast variable. So that's kind of the main takeaway here, but this is kind of a general model. Okay, and then so how does the width of this chair change with different parameters? So another parameter we had was the strength of the feedback, this alpha parameter. You can see in the equations down here. And what that does is it just varies the slope of the linear nullcline without changing the structure of the cubic nullcline. And as you increase the slope of the linear nullcline, what actually happens is you widen the chair. So this stronger feedback yields a wider seat of the chair. And you can characterize this or quantify this by looking at the two-parameter bifurcation diagram in input and feedback strength space, so J alpha space. So as you increase alpha, the distance between the subcritical Hopf bifurcation points increases linearly, and so the width of the chair will also increase linearly as you vary alpha. And so the width of the chair also depends on the system parameters, which is sort of intuitive as it should. And so that's a fairly general idea, but can we show this in a concrete biological example? So the model I'm going to talk about, the concrete biological model, it has to do with the islet of Langerhans and insulin secretion. So islets are part of your pancreas. They consist of about 500 cells, different types of cells, each cell producing a different type of hormone. And there's like a million islets in a human pancreas. Okay. And so in this sort of zoom in of a particular islet, you have alpha cells, beta cells, delta cells, epsilon, just a whole bunch of different types of cells. And all of these in some way are responsible for regulating glucose levels in your body. Um, and so, for example, if you're type 1 diabetic, what happens is your beta cells just die. Your beta cells are killed by your immune system, and maybe some of the other cells are too. I don't know for sure. But the way these islets and these beta cells work is very similar to neurons. So they're like synaptically coupled, and they secrete insulin in a very coordinated way. And so, with that in mind, Teresa Che and Joel Kaiser developed this Hodgkin-Huxley style model to describe insulin secretion and beta cell behavior, basically. So, the Hodgkin-Huxley type model for beta cells. And the key parameter here is this Kc parameter that shows in the equation for intracellular calcium ion concentration. And it's the rate at which calcium is pumped out of a beta cell. So you have two fast variables here, the membrane potential and the fraction of open potassium channels. And then you have a slow variable describing intracellular calcium ion concentration. This model exhibits a relaxation oscillation, but in particular, it has bursting behavior. So the membrane potential bursts, meaning you have sort of a limit cycle encoded within the separation of timescales that allows at the top part of this blue oscillation, you get like a faster oscillation at the top. Um, what I'm showing in panel A here is what's called the critical manifold. So if you see C is the slow variable. And so I'm treating C as a parameter. I look at what the null line affiliated with the membrane potential and potassium ion channels looks like. And that's this bifurcation diagram looking structure. And so you have a stable equilibrium, and then you have a Hopf bifurcation along the top branch. And then you have like a saddle node type of bifurcation that kills the oscillation. And then you have this bistable regime where you can jump between an oscillation and a stable equilibrium. And then the green triangles are two saddle node bifurcations. Um, and so if you superimpose a trajectory in the phase space with C varying slowly, you can see that the trajectory will track this null line. And then once it reaches the saddle node, it'll jump up to the limit cycle on the top branch of the critical manifold, and you'll see this oscillation until it reaches the saddle node of um, uh, of limit cycles on the upper branch, and then you come back down. So you have this jumping between these equilibrium states, where in one state you're oscillating. Okay. And so you find that if you measure the temporal average of the intracellular calcium concentration, you again see this sort of chair behavior emerging. So between the values of the pumping out rate of calcium, you get like a decrease in the calcium concentration. But once you start bursting, which is where you have this middle seat behavior, you maintain a relatively constant intracellular calcium concentration. And then if you increase the rate at which you pump out calcium too high, you lose the bursting and you start spiking tonically. But the point is that during this relaxation oscillation, bursting oscillation, the amount of calcium, intracellular calcium, is relatively constant. And this is important because calcium can trigger a lot of different things. It can induce, for example, apoptosis. It can trigger just a bunch of different things can go wrong if your calcium is not uh, regulated. And we're varying the rate at which calcium is pumped out, um, but the amount of intracellular calcium is actually relatively unchanging. And panel B, I'm showing the intersection of the C nullcline with the critical manifold. So each intersection of these colored curves with the Z critical curve is a true equilibrium of the system. And then you can see for what values or for which nullclines you can observe the homeostasis. And again, the fast variable does not show invariance because the duty cycle strikes again. The amount of time you spend in one phase or the other significantly varies the temporal average. So last question, what happens if you have multiple slow variables? Like what if you have a system, and we're going to look at what's called the phantom bursting model, which was developed by my collaborator here, Richard Bertram and Artie Sherman and Les Sattin. Um, there was a more detailed version of the Che-Kaiser model for beta cells, but you have three time scales. You have a fast membrane potential and a fast dynamics of potassium channels. You have a slow cytosolic calcium concentration and endoplasmic reticulum calcium concentration. And then you have a very, very slow time scale on which the ratio of ADP to ATP changes. So which variable, if I'm saying you have to measure the slow variable to observe this homeostasis, which variable do I look at in order to observe this homeostasis if it exists at all? Um, so in this example, I have A oscillating. A again is the concentration, is the ratio of the concentration of ADP to ATP. And the dashed lines show that the temporal average is not changing. So in this case, the ratio of ADP to ATP is driving the bursting oscillation. And so you have to measure that in order to see the homeostasis, the dynamic homeostasis in the model. The phase space representations at the bottom both show these bursting behaviors, but in the VC phase space, you have bursting, and then you kind of have this accumulation of bursting where you have this bursting happening and it kind of stays still. You have this accumulation. It's called phantom bursting. And it has to wait until A reaches the corresponding saddle node bifurcation to push it back down to the lower branch. And so you have to measure the slowest variable in this case because it's driving the bursting. But you can vary the conductance of the calcium activated potassium channels. And then if you really increase the conductance, then like your ratio of ADP to ATP does not change in time. And so in this case, it's the calcium ion concentration which is going to drive the emergent homeostasis. So you see that the temporal averages are not changing that much as you vary an input parameter. And so in this case, you have to measure the intercellular calcium ion concentration. And so the point is you always have to look at the slow variable, but it has to be the variable that drives the bursting in these examples or drives the relaxation oscillation. Um, so just to conclude, we've demonstrated like dynamic homeostasis and relaxation and bursting oscillators in multiple time scale oscillators. Looking at the slow variable will show invariance, and then when you have multiple time scales, you have to look at the variable that drives the bursting or the relaxation oscillation. One point that I did not talk about in this talk is that if you actually introduce stochasticity into the input parameters, then the seats of these chairs of homeostasis, the region, actually grows and widens. And so if you're interested in that and want to see more on that, please check out this paper. I'm very happy to say that this paper was the very first paper that was accepted in the new SciAm Journal on Life Sciences. So you can go read it. It's quite a lengthy paper, but and I think it's quite good. Acknowledgement to my student, Chris Rizovitz, who's done some really great work on this, and Richard Bertram for allowing us to analyze his model. Okay, thank you.
Well, let's thank Bhargav for a really interesting talk. Uh, so Sarah has put a question in the chat. So thank you very much for a very cool talk. In the calcium potassium channel example, can the plots of the VC without bursting be considered limit cycles at fixed points, or does the moving away from these fixed points lead to chaos or nonlinear dynamics?
Okay, so which, let me pull, like, oh, like in this slide? Oh, can you still see my slides? So you're asking, okay, in the calcium potassium channel example, can the plots of VC,
I can't see the slide anymore, actually, but
oh,
that might just be me.
okay, this one?
Oh, there it is.
Hmm.
Yep. No, but further in your talk, if you go further, these ones, like the ones in the bottom left, like without those bursting events.
Yeah, yeah. And okay, so I'm not able to see the chat and my slides for some reason. So can you repeat your question?
So I don't know if this is a well-posed question, so I apologize, but can you consider these like closed functions to be like limit cycles in some way? Without the
Yeah,
bursting, if the bursting wasn't there, and okay, so yes. Um,
yeah.
and if you move like so limits like so this is not my field, so uh you're at fixed points in your dynamical system. If you move away from those fixed points, will you get to like chaos, like some sort of chaotic nonlinear situation?
Not in not in this model. I don't know in general, but what's happening in this model is as I vary a parameter, you have a stable fixed point um, that undergoes a Hopf bifurcation. And so you have a Hopf bifurcation on sort of the slow time scale. And so if you just look at the slow time scale, all you'll see is like you'll see like a standard looking limit cycle. But then you have to look at the fast time scale because on within that slow limit cycle, you have another limit cycle superimposed on top. And that occurs only on the top branch. And so this is like the bursting. So, I mean, I don't think you see chaos in this system. This is just kind of like limit cycle superimposed upon limit cycles.
Yeah, so Zonia has put in the chat there are other neural models that can exhibit chaos for some parameter ranges.
Okay,
Oh,
thank you.
Yeah. So I have a question for Bhargav. I think it's really, really cool to think about, this dynamic homeostasis. Um, so I'm just wondering, like, if you compare this dynamic homeostasis in this relaxation oscillation systems, you know, does it maybe provide extra stability, uh, compared to maybe the more classical, just, steady state homeostasis systems? I'm actually thinking in my mind an analogy like, people riding a bike. It will be, very difficult to stay balanced if you're not, moving the bike, right? So, but once the wheels start rolling, it's much easier. It's probably not in a very appropriate analogy, but can you do some maybe comparison between dynamic homeostasis and the classical steady state homeostasis?
yeah, I think that hits actually right on the spot is that these uh okay, so first of all, I guess it depends on how you like impart variability. Like if you look at it from a stochastic perspective, it depends on how you impart the stochasticity. Um, if you do like additive Brownian motion to the fixed equilibrium, then like a fixed equilibrium, fixed homeostasis, static homeostasis example, then it doesn't really do anything. You just average out the noise, and the static equilibrium is pretty stable. It's pretty robust. But what you don't see in like these static homeostasis models is when you add noise, the region kind of doesn't change and sometimes shrinks in terms of the homeostatic region. But when you have these dynamic homeostasis, the regions on which you actually see this homeostasis increases quite drastically, because noise can push you across bifurcation points And
Mm,
so one of the selling points that we had is that you have like a more robust, like it's a more robust mechanism to encode
mm,
homeostasis than the static one. Yeah. That's a good
that's
point.
super
I
cool.
I think the cycle is a cool example. Yeah.